I'm Peter Cheung from the School of Public Health. My broad interest has been to understand the genetic diversity of influenza A viruses. Genetic diversity means that upon infection, after every round of virus replicating, there are mutations or genetic changes that occur in the genome, thereby formulating a population of genetically dissimilar genetic variants. And the reason for these mutations is due to an enzyme called the polymerase, which is responsible for the copying of the genetic blueprint in the influenza A virus genome. And the consequence of that is that instead of having a genetically identical viruses in the population, we observe that in this population there are viruses that differ by one or a few mutations. Our laboratory aimed to study genetic diversity using different approaches. The first is to use bioinformatics or computational methods to identify whether there are specific patterns or properties in the mutational spectrum or landscape of this influenza A virus's genome. Our second approach is to develop experimental models to understand the biological significance of genetic diversity during influenza A virus adaptation in the host. This paper provides experimental evidence for the role of genetic diversity in influenza A virus survival. And to achieve these, we look at the polymerase closely, and we randomly generate mutation or changes in the amino acids to see whether we can alter the accuracy of the polymerase. And a more accurate polymerase conceivably would lead to a decreased genetic diversity once this virus begins to replicate. And we found that one mutation in the PP1 gene of the polymerase was sufficient to change the accuracy of the polymerase, leading to a restricted genetic diversity. And we examined biological properties of these mutant viruses. We observed that restricted genetic diversity leads to a decreased ability to adapt to the changing environmental conditions in both tissue culture and in mouse model. In this study, in order for us to generate random mutations in the polymerase that can alter the accuracy of the enzyme, we've used a mutagen. And we show in this study that causing mutagenesis in the influenza A genome can actually be an antiviral approach in that by generating a lot of random mutations in the viral genome, the virus would suffer a lot of deleterious mutations that can cause the virus to collapse. And the second point of this study was that we have introduced a new mechanism for influenza A virus virulence. By generating a virus with a decreased ability to generate genetic diversity, we show that the genetic diversity can also be important for the virus to adapt to the changing environment within the mammalian host. And there are two approaches as a third extension to this study. To cause mutations or mutagenesis in the influenza A genome by a mutagen can be further explored as a viable treatment option to inhibit influenza A virus growth. And influenza A viruses has a segmented genome in that there are separate copies of these gene segments that can be shuffled when two dissimilar viruses infect the same cell. And this is one way to obtain genetic diversity. So it is not known whether just random mutation alone is important for the virus to adapt to the changing environment. And we provide the first experimental evidence that indeed genetic diversity generated by random mutations is very important for the virus to survive. And we hope that by having these mutant viruses with decreased genetic diversity, we can further explore its importance in not only antigenic drift, antiviral resistance, tissue tropism, but also transmission among different hosts.